My name's Kelly Turkington. I'm a plant pathologist with Agriculture and Agri-Food Canada in Lacombe, Alberta. Originally from Saskatchewan, and I work on cereal and canola diseases. So this particular trial is part of the canola cluster program right now, and it's looking at uh, evaluating risk assessment tools and how that relates to weather conditions, crop canopy environment, and ultimately response in terms of fungicide timing. So the trial itself has two seeding rates. We're looking at 60 and 120 seeds per meter squared, and then 9 fungicide treatments. So we start with a check treatment. We have what is called the yellow bud treatment. That's sort of our anchor treatment. And it's based on some concepts and ideas from the United Kingdom. So they're looking at um, fungicide application at the yellow bud stage, and the focus there is more to get the fungicide down into the crop canopy, down where the key infections that cause the most severe yield losses. And in some cases, depending on the particular product they're using, it may have a bit of a benefit in terms of plant growth regulation and lodging. So that's our anchor point treatment. So then we have a yellow bud treatment, a single, or we wait and come in with a single application a week after yellow bud, or 2 weeks after yellow bud, or 3 weeks or 4 weeks after yellow bud. So those are all single applications starting very early and very late. And then we look at 3 dual applications. So the anchor point, the yellow bud application, and then we come back in with the second application at 2 weeks after yellow bud, or 3 weeks after yellow bud, or 4 weeks after yellow bud. So layered on top of that is we're measuring spore load in the air or on the petals. And the importance of assessing spore load relates to the disease cycle of Sclerotinia. So if you look at Sclerotinia and its disease cycle, the pathogen, and if you can imagine at the soil surface in mid-June when the crop canopy covers that soil surface, the sclerotia that are in the soil, close to the soil surface or at the soil surface, will start to germinate. And they'll produce apothecia with those little golf tee-like structures. And those apothecia will release ascospores into the air. So those ascospores will land on canola petals while they're still in the flower. And those infested canola petals will then drop down and lodge in the crop canopy. So they'll lodge in leaves, leaf axils, leaf bases, and often you'll get clumping of petals, you'll get nice big juicy water droplets, so an ideal environment for infection. So in the case of Sclerotinia, that's one cycle. So spore production, release, deposition on the petals, infested petals fall down, the fungus uses the petals as a carbon and nitrogen source. So when you look at the end of the season and final disease levels, the amount of disease at the end of the season will be proportional to the amount of inoculum, ascospores floating around in the air during the flowering period. So with Sclerotinia, a producer or a crop consultant advising the producer needs to make that call to spray or not to spray before they actually see disease symptoms in the crop. In the case of the trial that we're looking at the different timings, starting at yellow bud and then 1 to 4 weeks later, or dual applications. But we're also monitoring spore levels in the air. We're doing that with a number of different technologies. So we're using a research-focused spore trap, which is the rotorod trap, and it's a trap where you have silicone-coated rods which spin around at high speeds, 2400 RPM. Spores are impacted on the rods, and then we take the rods and test the rods for DNA. And the other thing that we're doing is we're collecting petals to assess the amount of Sclerotinia DNA on the petals. Now, if we look at this type of approach, and we look at the nature of Sclerotinia, and again, the fact that it's a monocyclic disease, the one component of the disease triangle, so the host, the pathogen, and the environment, that is quite important for Sclerotinia is assessing spore load, either just prior to flowering or during the flowering period. 2019, we had trials across Canada and including here at Outlook. And in that year, most of the sites had relatively low Sclerotinia, and we had some sites that had higher levels of Sclerotinia. And that was at Beaver Lodge and here at Outlook. Outlook was fairly low, Beaver Lodge a little higher. And then we had a master's student, Eleanor McBain, who worked out of the University of Alberta, and she looked at commercial fields in that Edmonton region. And Eleanor looked at doing very much the same thing that we're doing here in terms of monitoring spore load. Also, she was looking at relative humidity and temperature both above the crop canopy, so ambient levels, and within the temperature both above the crop canopy, so ambient levels, and within the crop canopy. And Eleanor had very high levels in the fields that she surveyed in 2019, I think in that range of probably 40 to 70% plant infection, so significant yield losses in those fields. 2020, she conducted her trial, and she had lower levels of disease, maybe in around that 20%. But overall, if you look at the results from 2019 from our fungicide trial, including here at Outlook and at Beaver Lodge and Eleanor's work, and she was doing the same type of testing that I just mentioned. What we found is that in general, the amount of disease at the end of the season was significantly related to the amount of inoculum during the flowering period. Through Eleanor's work, she demonstrated that, and our companion fungicide trials, that there's a threshold. If you look at quantitative PCR and the test that Barb originally developed, whether you do it on petals or the rotorot samples, is that it seems like there's a threshold of 0.0001 nanograms of Sclerotinia DNA per petal or per unit volume of air per unit of time. And sites that had little to no disease had much lower levels of DNA based on the rotorod sample and the petal test that we were doing. And the testing that was done in conjunction with Dr. G. Fang, who was with the Alberta Plant Health Lab in Edmonton. The other aspect of Sclerotinia, though, is that although disease at the end of the season is proportional to the amount of inoculum floating around during the flowering period, that relationship is very strongly influenced by weather and the crop. So in the case of weather, it's primarily moisture that's driving that. Temperature can have a moderating effect, but it's primarily moisture. And so relative humidity is a key parameter there. The crop itself can have an impact in terms of crop development, interaction with weather conditions, occurrence of rainfall, and so on. But also the crop will have an impact in terms of crop canopy density. And that, to me, the easiest way of looking at that is yield potential. So if you have a relatively low yield potential, let's say your yield potential is well below 40 bushels per acre, you probably have a fairly light crop canopy. It's not going to produce conditions within that canopy that favor disease. And as a consequence, you're going to have limited risk and likely not a need to spray fungicide. But once you start getting above 40, and when I did all my work a long time ago, we were quite happy with a 40 to 45 bushel per acre crop. 45 was probably outstanding. Now with the new hybrids that we've had over the last probably 15 to 20 years, we're looking at 70, 75 bushels per acre, perhaps even higher. So as you get that denser crop canopy, it's going to hold the moisture in for a longer period of time. So you have longer periods of leaf wetness, longer periods of high relative humidity. So in terms of the RH, that was a key parameter. And what Eleanor found, and we found in our companion fungicide trials, is that those sites that had little to no Sclerotinia had really, during the flowering period, average RH was below 80%, and in some cases well below 80%. That if you look at the disease cycle, you've got sclerotia in the soil, you get germination of the sclerotia, production of apothecia, release of spores. So we're getting closer and closer in that disease cycle to the actual occurrence of disease. So if we look at aerial assessment of spore load, that level of spores that you see at that particular time is a reflection of the amount of sclerotia that are in the soil or in your field or adjacent field, and the favorability of the weather. So it kind of synthesizes that. If you think of the disease triangle, measuring that inoculum level is accounting for the impact of weather and crop development and so on. Now, if you test petals and the amount of DNA, that's one step closer in the disease cycle to the actual occurrence of disease. So certainly in Sclerotinia, I think it's very important to look at all, and that's certainly what Eleanor concluded in her master's thesis, all components of the disease cycle, including the pathogen. So again, this is the last year of this particular project, so we're hoping next spring to have some better set of guidelines in terms of producers and crop consultants in terms of assessing Sclerotinia risk, timing the fungicide application, and improving the ability to make some well-informed decisions in terms of fungicide use.